Spiroligomer. Spiroligomers, also known as bispeptides, are synthetic oligomers made by coupling pairs of bis amino acids into a fused ring system. Spiroligomers are rich in stereochemistry and functionality because of the variety of bis amino acids that are capable of being incorporated during synthesis. Due to the rigidity of the fused ring system, the three-dimensional shape of a spiroligomer, as well as the display of any functional groups, can be predicted, allowing for molecular modeling and dynamics. Synthesis. Spiroligomers are synthesized in a stepwise approach by adding a single bis amino acid at each stage of the synthesis. This stepwise elongation allows for complete control of the stereochemistry, as any bis amino acid can be incorporated to allow for elongation, or any monoamino acid can be added to terminate a chain. This can be accomplished using either solution phase or solid phase reactions. The original synthesis of spiroligomers allowed for functionalization on the ends of the oligomers, but it did not allow for the incorporation of functionality on the interior diketopiperazine (DKP) nitrogens. Much work has been done to allow for the functionalization of the entire spiroligomer, as opposed to just the ends. By exploiting a neighboring group effect, spiroligomers can be synthesized with a variety of functional groups along the length of the molecule. Structure. Spiroligomers can be synthesized in any direction and between any pair of bis amino acids. Spiroligomer diketopiperazines can be created between either end of a bis amino acid. Spiroligomers are known to be conformationally rigid due to the fused ring backbone. Chemical characteristics. Spiroligomers are peptidomimetics, completely resistant to proteases and not likely to raise an immune response. Uses. Spiroligomers have been utilized for a variety of applications which include catalysis, protein binding, metal binding, molecular scaffolds, and charge transfer studies et al. Catalysis. Two unique types of spiroligomer catalysts, spiroligozymes, have been developed, an esterase mimic and a Claisen catalyst, transesterification. The first spiroligomer catalyst was an esterase mimic, which catalyzed the transfer of a trifluoroacetate group, aromatic Claisen rearrangement. The second spiroligomer catalyst accelerated an aromatic Claisen rearrangement with a catalytic dyad similar to that found in ketosteroid isomerase, protein binding. A spiroligomer was designed to mimic p53 and bind HDM2. The molecule enters cells through passive diffusion, and this mimic was shown to stabilize HDM2 in cell culture metal binding by nuclear metal binding molecular scaffolds, rods used for distance measuring with spin probes, electron transfer, donor bridge acceptor. Other uses. Possible applications that are currently investigated include the binding and inactivation of cholera toxin and the cross-linkage of surface proteins of various viruses, such as HIV and Ebola virus. Further, the group of Christian Schaffmeister developed molecular hinges, which can be used for the construction of molecular machines, such as nanovalves or data storage systems. See also molecular engineering, molecular machine, molecular nanotechnology, and nanotechnology.